So unless you've done a single cell transplant with one cell showing that it has become everything, you haven't shown that adult cells have any degree of plasticity or flexibility at all. So that was the challenge. And there aren't a lot of people who can do single-cell bone marrow transplants in the world. There's about a dozen of them. But one of them, Saul Sharkis, is a colleague of Diane's and down at Johns Hopkins. And he was in the process of actually doing those experiments. He had done them 5 months before. Out of 30 mice he transplanted with a single cell. I'm not going to go into the details of how he derived that special cell. It's a functional assay, not by markers or anything, but a single cell, not a single cell type, but one cell, rescued 6 out of 30 mice that were transplanted with it who got lethal irradiation. And we knew that 5 months hence, he was planning on sacrificing those 6 animals, and we asked him if we could have all the tissues when he was done taking out the spleen and the bone marrow and the blood to look at. And he said yes. So we did, sort of like, waiting for mice to be killed, descending on Baltimore with my bottles of formalin. And that's a very unpleasant story meant for parties when you've been drinking. But invite me back. It's very entertaining, but not with your faculty here. So it's lovely being in a room full of students. So we looked at these tissues, and once again, we saw hepatocytes from the bone marrow. We were not surprised by this. We also found bile ducts from the bone marrow, and the colors will change from slide to slide because of changes in filters. Here, the Y chromosome is blue. All the yellow, orange, or brown, or red staining you'll see are cytokeratin. We did co-staining for immunostaining and FISH for Y chromosome and keratin to define epithelial cells. These are Y chromosomes in the bronchial lining. These are Y chromosomes in the type 1 and type 2 pneumocytes. And type 2 pneumocytes, which we prove using other stains, are the stem cell, intra-organ stem cell for the lung. So lung stem cells in the lung coming from the bone marrow. Esophagus. Where is it? There it is. Stomach and small intestine. Crypts of the colon. Again, that's the location for the intra-organ stem cell compartment in the colon, but it's coming from the bone marrow. Autofluorescent hair follicle, hair shaft and a hair follicle. The epidermis is up here, and this is the bulge region of the hair follicle, the intra-organ stem cell compartment for the skin, but it's coming from the bone marrow. So we had one adult cell reconstituting all the blood elements in the bone marrow and the circulating blood supply, epithelia of all the viscera, skin and hair follicles, and that meant we had one cell giving rise to endoderm, ectoderm, and mesodermal tissues. And so this was the first true clonality experiment that proved adult stem cell plasticity. If you remember when the stem cell debate started, it was 2001. It was the first week in May in 2001 when the anti-abortion lobby started saying, we don't need to do embryonic stem cells because adult cells can do everything. And then the debate exploded. It was in response to this paper, which we published in Cell in the first week of May 2001. So I thought, now can I go back to liver pathology? But no. So the problem with this is that one of the reasons, aside from the politics, that people were aggressively resistant to this is because we have this kind of standard model of how cells develop, either through embryologic development to postnatal tissues, or in terms of maintaining tissues in response to injury or just general growth. And so we have stem cells which divide, self-renew, give rise to progenitor cells, which then divide. Some of those self-renew. And as you move down from left to right, everything in a unidirectional hierarchical fashion proceeds in order. And this was established by cell transplant experiments in frogs, initially going back 100 years ago. When the molecular era opened, they started identifying that there are irreversible gene restrictions that take place at each of these junctures so that cells can only go forward, they can't go back. And that's probably what you've been taught, yes? Is that your embryology? Have you done embryology yet? Yeah, okay, and they taught you this, right? Well, they're wrong. First, the question is, well, what if gene restrictions are reversible? And it turns out that in the last 10 years, parallel with all this plasticity data, the field of irreversible gene restrictions, we know what the mechanisms for that are, and think of the most extreme is exon activation. But there are more subtle things with heterochromatin formation, et cetera. It turns out at first, experimentally, all of those could be reversed, but now we know physiologically all of these can undergo reversal. And if that's true, then we have to think that there are other pathways we can find if we look for them. And sure enough, people have demonstrated cells going backwards along these pathways. This is the kind of experiment Diane and Saul and I were doing. Early progenitors go back to embryonic-like stem cells. And here we have something like a terminally differentiated cell, and that's our language for it, a terminally differentiated cell. Let's say a cell of a mammary gland could, as a random example, could actually become something like an embryonic stem cell. And the proof of principle for that is Dolly. Because if you don't know, Dolly was actually cloned from a mammary duct cell, and that's why she's called Dolly, because she's named after Dolly Parton. When I got to visit Dolly, but the other part of that story is that the people who, at the institute where Dolly was made and was born, the techs on staff at the time an animal is born from one of their experiments gets to name the animal. And the tech on staff was a huge Dolly Parton fan. And so they had been talking, what would we call the creature derived from this breast cell? And someone joked Dolly Parton, and he thought, if I'm on call, I'll name her Dolly, and Dolly Parton will come visit the first clone. And sure enough, Dolly did come to visit the first clone. That's why Dolly doesn't have any wool, because a few months earlier, they had her shorn to make wool into yarn to knit a sweater for Dolly Parton on her visit. And then Dolly autographed it across her cleavage, and they auctioned it off for a local children's hospital and made $20,000. So that's why some people see this picture and go, what are you doing with a baby cow? It's like, it's Dolly. So Diane, when she talks about this stuff, likes to use this image. We have all these dogmas we're trained as being really rigid and inviolable, and you have to beware of dogma, because even though the dog is sleeping, you wake it up, it can really bite. We'll come back to this image. So thinking about this critically, Diane and I started thinking, well, what does this mean in terms of the biology we've been taught and about what cells are capable of? And so we published a series of 3 papers on what we called 3 new principles of plasticity. My favorite and the fullest version of that was published on April Fool's Day in 2002. And so we described 3 principles. I'm not going to go into them in great detail. Genomic completeness simply means that if a cell has the entire genome intact, it's not 100% necessary. But if it has it, then it can theoretically become any cell type. And cloning is a proof of that principle. And now we've got, if you read in the New York Times, they're always talking these days about induced pluripotent stem cells when they talk about stem cell news in the Tuesday Science Times. It's a further proof that this is the case. What I want to focus on here is cellular uncertainty. And I called it cellular uncertainty because I was trying to echo Heisenberg's uncertainty principle. Presumably, if you're all sitting here, you had to do physics at some point? Would you like me to briefly explain Heisenberg's uncertainty principle? Should I review that? OK. Heisenberg said at some point in the 1940s, I think it was, or maybe 30s, that any attempt to observe a subatomic particle to figure out its momentum or its location or its speed or its direction, anything about it, you'd have to fire some sort of energy beam at it made of photons. And those photons are going to hit the particle. And while you may be able to figure out precisely where it's located, you've now changed its momentum or its direction. And if you figure out a way to figure out its direction and momentum, you're going to have, with great accuracy, then you'll screw up where it's located. And so you can't figure everything out about a particle. Initially, he actually said in the first publication, maybe this is a technological limitation, and we could surpass that. But as quantum physics developed, within a few short years, they realized, no, this is a property of matter at the subatomic level. That's why we talk about electron clouds as opposed to points orbiting around an atom the way we would classically draw an atom. But you have electron shells that are cloudy because of uncertainty. So I was referring to Heisenberg's uncertainty principle here mostly to be provocative, which you've probably gotten a sense I kind of enjoy being provocative. What I was basically restating is another dogma that we already know, but cell biologists tend to ignore all the time. Richard Lewontin put it very nicely. The internal and the external co-determine the cell. So in the genomic era, we know that the genome is a powerful drive for determining what a cell is going to be. But it's not just the genomic expression, it's also the impact of the microenvironment on the cell. And the nature of a cell arises where the inside meets the outside, and they interact with each other, and then a cell becomes what you see under the microscope or in a culture dish, et cetera. So what we wrote for the second principle was any attempt to observe a cell alters the state of that cell at the time of characterization and potentially alters the likelihood of subsequent differentiation events. So it's like Heisenberg, but at the cellular level. We said potentially because, like Heisenberg initially did, we were saying, you know, maybe one day we'll have a perfect MRI machine, and we'll be able to tell everything about a cell without disturbing it in any way. And so we weren't saying this is a property of cells. We put that caveat in there, and we'll come back to that. So meanwhile, we're writing about this and having a good time, but the plasticity debates are now raging because of the adult versus embryonic stem cell stuff, and we were in the thick of this, unfortunately. So now, because it was perceived that to support embryonic stem cells, you had to suppress the adult stem cell research, which was very much active at the funding levels, at the publication levels. Our paper in Cell, they actually offered us one of the reviewers, who's a member of the National Academy of Sciences and wrote the National Academy's report on stem cells for the government. He reviewed our Cell paper. And he said in his final review, it took 8 months to get it published, and the senior editor of Cell quoted him in her acceptance letter to us, saying, each of the authors should be keenly aware that for each of them, this is a quote, I've memorized it, for each of them, this paper can be a reputation maker or a reputation breaker. Close quote, and then offered us the chance to withdraw the paper rather than publish it. So this is what stem cell research has been like, even in the adult world, which theoretically has universal support. It has not. So having answered the single cell question, the next argument against adult stem cells, well, you showed it happen, but most of the cells I showed you were 1 in 10,000, 1 in 100,000, 1 in a million cells in the liver or the skin or the brain, et cetera. So unless you show that it's truly robust, it's trivial. It doesn't really matter, so who cares? Which ignores two things. One is if it happens 1 in a million, you can still figure out how it happened and manipulate that for therapeutic purposes. So wouldn't that be a good thing to do? But it also ignores the fact that in the lungs of these mice, they had radiation pneumonitis, not lethal, but severe. And in the lungs, 20% of the pneumocytes were derived from the bone marrow. So we had already showed robust engraftment. If the kind of injury and severity of injury was right, you actually got robust engraftment. But this was just, and in a human liver, we showed, we were the first ones to show this kind of thing happening in humans. And in one post-liver transplant hepatitis C, 40% of the patient's liver had reconstituted from his own bone marrow. He had received a female liver, so we could use his Y chromosomes to track. So it had been shown. But this didn't really get at some fundamental principle, and I thought there had to be a better answer to that, which I didn't get until I met this artist named Jane Prophet. And Jane is a new media artist. You should go online and Google her. She does really nifty stuff, and a lot of it's well documented online. And now she's in New Hampshire, but she used to be at the University of Westminster, where we have a mutual friend whose job it is to curate art-science collaborative interactions, because in Europe, they really think a much more interesting way to develop creative solutions to problems is to get people from different disciplines talking, and then you get more creative solutions. So they want to see how to foster that. And so he actually, and he's wanted to introduce us for about 10 years, and we were never in the same city at the same time.